students, welcome to the session. So in this session, we are going to discuss a sample question paper of biology. Okay, so before moving on to the questions, let us see some of the instructions. Okay, so all questions are compulsory and the question paper has 5 sections and total there are 33 questions and all questions are compulsory, right? Then section A has 16 questions of 1 mark each. So it may be from one word answer or you should answer in a simple words, okay? And section B has 5 questions of 2 marks each. Section C has 7 questions of 3 marks and section D has 2 case-based questions of 4 marks each and section E has 3 questions of 5 marks each. Okay, so there are certain internal choices there. There is no overall choice, but there are some internal choices which have been provided in some questions and the student has to attempt only one of the alternatives in such questions. And don't forget, wherever necessary, neat and properly labeled diagrams should be drawn. Okay, so let us move on to the questions. And the first question from section A is, an infertile couple was advised to undergo in vitro fertilization by the doctor. Out of the options given below, select the correct stage for transfer to the fallopian tube for successful results. So, what is there in the question? An infertile couple has been advised by doctor to go for in vitro fertilization. So, they may be having some kind of problems in conceiving. So, see here some options are given there and we know that in case of in vitro fertilization, the fertilization is taking place outside a female body, right? And once the zygote is formed, it has to be transferred into the woman's body, right? So, if we are transferring an early stage of embryo, we should go for the fallopian tube, right? Because from the fallopian tube towards, when the zygote is moving towards the uterus, it is undergoing certain kind of changes. Okay, the single cell zygote will undergo cleavage to form 2-celled stage, then it will change into a 4-cell stage, then 8-cell stage. Okay, and after that a mulberry-like structure will be formed which is known as morula. After that, it is then getting converted into blastocyst where a rearrangement of cells is going on there. Right, where the outer layer is known as the trophoblast and the inner cell mass will be there and that particular blastocyst is getting implanted into the uterus. So, any stage of this embryo zygote till the 8-cell stage, it has to be transferred to the fallopian tube. Then the late embryos, the advanced embryos has to be transported into the uterus. So, let us see the options here. Option A, zygote only. Option B, zygote or early embryo up to 8 blastomeres. C, embryos with more than 8 blastomeres. And option D, blastocyst stage. So, the right answer is option B, zygote or early embryo up to 8 blastomeres. After that, that means after the cell has reached to a stage where it has more than 8 blastomeres, where blastomeres are individual cells, right? So, if these like zygote early embryo is having more than 8 blastomeres, it has to be transported into the uterus. Okay. So, the right option is option B. Then, next, given below are 4 contraceptive methods and their modes of action. Select the correct match. So, the options are C, A, condom, option B, vasectomy, option C, pill and option D, tubectomy. And see here mode of action, the first one is ovum, not able to reach fallopian tube. Then option 2, prevents ovulation. Option 3, prevents sperm reaching the cervix and option 4, semen contains no sperms. Okay, so let us see. The first one is the condom, right, where it prevents the sperm from reaching the cervix. So, we can say that A, it will match with 3, right. Then vasectomy. Vasectomy is actually a surgical method which is used in males. It is a terminal method. right? So, vasectomy, it prevents, here semen will contain no sperm, right? So, B will go with 4. Okay, then C, pill, where pills, they basically prevents ovulation. So, we can say that C will go with 2. Then D, tubectomy, here it is a surgical method used in case of females where the ovum will not be able to reach the fallopian tube. So, we can say that D will go with 1. So, the right option is A 3, then B, here 4, then C 2 and D 1. So, the right option is option C, A 3, B 4, then C 2 and D 1. Okay. So, let us move on to the next question. Then, Apis mellifera are killer bees possessing toxic bee venom. Identify the treatment and type of immunity developed from the given table to treat a person against the venom of this bee. Suppose this bee has sting a person. So we don't have enough time to wait for our immune system to identify that particular venom and to actively produce antibodies against that, isn't it? So, we will definitely go for passive immunity. So, because we don't have enough time to wait for our immune system to act at that situation. So, we'll go for passive immunity only, right? So, let us see the remedy. Inactivated proteins, active immunity. No, we will not go for active immunity because we don't have enough time, right? Then, proteins of the venom and passive immunity. No. If we are injecting proteins of that particular venom, we are making our immune system to identify and to act against that. So, it is not an active, passive form. It is definitely an active form, right? So, this is not the right option. Then option C, preformed antibodies, passive immunity. Definitely, preformed antibodies are providing passive immunity and we will be going for option C only. So, the last option is dead microorganisms. It is of no use. It is not applicable in such a situation. So, we are going for option C only where we will be using preformed antibodies which give us passive immunity. Okay. So, next question. Interferons are most effective in making non-infected cells resistant against the spread of which of the following diseases in humans. So, we know that interferons are produced by cells which are attacked by, affected by viruses. Okay. So, if this is a cell which is infected by a virus, it will start producing the interferons. It will start producing the interferons to warn the surrounding cells, of this virus attack. Okay, so basically these interferons will protect. It is actually aimed in protecting or warning the surrounding cells about this viral attack. So, among this option, which is a viral disease? See, option A, Ascariasis, it is a worm disease, ringworm, it is a fungal disease, amoebiasis, it is a protozoan disease, and AIDS. We know that AIDS is caused by human immunodeficiency virus, right? HIV virus. So, definitely the answer is option D only, AIDS. So, next we have an assertion and reasoning type of question. So, here it will consist of two statements, an assertion and a reason. So, we need to answer the question according to the options given below. So, if both A, that means assertion, and reason are true statements and the reason which is provided is a correct explanation of that assertion, then we should go for option A. If both assertion and reason are true, but we can't say that the reason is not the correct explanation. The reason is, if the reason is not the correct explanation for A, then we should go for B only. Then, if the assertion is true, but the reason is false, we need to go with option C. Then, if assertion is false, but the reason is true, we should go with option D. Okay. So, let us see the question here. Assertion, when white-eyed, yellow-bodied Drosophila females were hybridized with red-eyed, brown-bodied males and F1 progeny was intercrossed, the F2 ratio deviated from 9:3:3:1. This is a Mendelian ratio, dihybrid ratio, right? So, when a female like a Drosophila was crossed with, which is having a white-eyed yellow body, okay, which was white-eyed and yellow-bodied. When it was crossed with a red-eyed brown-bodied male, we will definitely wait for an Mendelian dihybrid ratio, but it deviated from that. And what was the reason for that? What was the reason T.H. Morgan gave for that? It was linkage, right? Linkage between genes, adjacent genes, right? Linkage between adjacent genes. If certain genes are present in a single chromosome, in the same chromosome, see, if this is a gene A and this is gene B, if they are closely located, they will exhibit some kind of linkage with each other, so that whenever they are forming a gamete, they will try to be together. Okay, they'll be inherited together, and we will get parental combinations in more numbers. Okay, so reason, let us see the reason. When two genes in a dihybrid are on the same chromosome, like I said here, the proportion of the parental gene combination is much higher than the non-parental type. Recombination will be lesser there. Okay, more the linkage, lesser will be the recombination. So this is the right explanation, right? So we can say this, the assertion and reason is true. Both the assertion and the reason are true, and the reason given is the correct explanation for the assertion there. So we can go for option A. Okay, option A. Then next question. Let us see the question from part 2. Here in the figure given below, parts A and B show the level of hormones which influence the menstrual cycle. Okay, study the figure and answer the questions that follow. So this is the figure where you can see part A is showing FSH. The FSH means follicle stimulating hormone, and LH luteinizing hormone. Okay, and here part B is showing estrogen and progesterone. Okay, so let us see the first question. Name the organs which secrete the hormones represented in parts A and B. So we said that part A was having FSH and LH. FSH and LH. And we know that FSH and LH are secreted by which gland? Which gland? It is secreted by pituitary gland. Pituitary gland. So these are the pituitary hormones. FSH and LH are pituitary hormones. They are collectively known as what? They are collectively known as gonadotropins. Gonadotropins. Then part B was showing which hormones? Part B was showing estrogen and progesterone, right? Estrogen and progesterone. So these are secreted by our ovary, right? These are ovarian hormones. So these are secreted by ovary only. Okay, so this is the answer of question A. Okay, then B, state the impact of the hormones in part B on the uterus of the human female during 6 to 15 days of menstrual cycle. So, during the menstrual cycle, we know that the first 5 days is the menstrual phase where bleeding occurs, and after that, what happens then? The proliferation of endometrium takes place there. So, they are basically helping in the proliferation of endometrium to welcome another pregnancy, right? Proliferation of endometrium, and it is basically done by due to the impact of this hormones from the part B. That means the estrogen and the progesterone. Estrogen is mainly helping in the proliferation of the endometrium. Okay, so let us move on to the next question. A true breeding pea plant, homozygous dominant 4 inflated green pod, is crossed with another pea plant with constricted yellow pods. Okay, with the help of a Punnett square, show the above clues and mention the results obtained phenotypically and genotypically in F1 generation. So, let us write the parents here. The parents are, see, one is homozygous. True breeding means they are homozygous, right? True breeding, homozygous dominant for inflated green pods. So, here constricted yellow pods are represented by small letters, right? So, inflated we can use capital letters because they are the dominant ones. So, here inflated, capital F, and green pods, capital G, inflated green pods, right? And then the other was constricted yellow pods, okay, small f, small g. And the gametes will be, see, the gametes are here, only one kind of gamete will be produced. So, we can represent it like this, Fg, right, Fg, only one kind will be produced here, Fg, here also Fg, okay, and here see, small f, small g, and again small f, small g, right. Then, what is the question here? With the help of a Punnett square, show the above crows and mention the results obtained phenotypically and genotypically. So, let us draw the Punnett square here. I am drawing it here. So, see. One of the parent, let us write the gametes here, capital F, capital G. Here also capital F, capital G. And here small f, small g. And here also small f, small g. Okay. So, the resultants are, resulters capital F, capital G, small g. Here also capital F, capital G, small g. Here also capital F, capital G, small g. And again capital F, capital G, small g. All will be of what? Inflated green pods. Okay, all of the offsprings are having what kind of phenotype? Inflated green pod. The phenotype, right, the phenotype. And what is the genotype here? The genotype is capital F, capital G, and small g. Genotype. Clear? Right. So, all are having inflated green pods only. Okay, then next question. During a field trip, one of your friend in the group suddenly became unwell. She started sneezing and had trouble in breathing. Name and explain the term associated with such sudden responses. What would the doctor recommend for relief? So, what will be the reason for her sudden change in physical condition? She started sneezing and had trouble in breathing. So, definitely it is indicating some kind of allergy, right? It is some kind of allergy where we can say that allergy is an exaggerated response of our immune system, right? It is an exaggerated, our immune system is making some kind of drama there, right? So, it is an exaggerated response by the immune system. It is playing some kind of drama, right? It is the exaggerated response our immune system towards some or any environmental factors, we can say. Any environmental factors like pollen, then dust, okay, dust, temperature, etc. Environmental factors. Okay, clear. So, what does the doctor recommend for relief? The doctor must recommend, he should recommend some kind of antihistamines or anti-allergens. Antihistamines Then some may recommend adrenaline, etc. etc. Okay, clear. So, let us see the next question. Answer the question from 22 to 28 and each carries 3 marks. Okay, so explain the functions of the following structures. Explain the functions of the following structures in the human male reproductive system. Okay, human male reproductive system. So, the first one is scrotum. Where is the scrotum located? It is actually located outside the abdominal cavity. Isn't it? It is located outside the abdominal cavity. And what is a scrotum? It is a pouch-like, a sac-like structure, isn't it? A pouch or sac-like structure. So, we can write a pouch-like structure which holds the testis, right? Which holds the testis outside the abdominal cavity. We can say it is a pouch-like structure which holds the testis outside the abdominal cavity. And what is the purpose of that? It helps in maintaining temperature that is needed for spermatogenesis. We know that spermatogenesis requires a temperature which is lower than our body temperature. And our body temperature is somewhat 37.5 degrees Celsius, right? Where spermatogenesis only requires a temperature approximately equal to 35 degrees Celsius. So, in order to maintain this much temperature, lower temperature there, the testis is actually located inside the scrotum. So, it helps in maintaining temperature, maintaining a lower temperature or temperature that is required for spermatogenesis. That is required for spermatogenesis. Okay, that is required for spermatogenesis. Then, so this was answer of the question number A. Okay, then what is B? Leydig cells. What is the function of Leydig cells? Where can we see these Leydig cells? So, B, it is the Leydig cells, right? Leydig cells are seen in between the seminiferous tubules. If this is a seminiferous tubule inside the testis and this is the second one, and if this is the third one, you can see the Leydig cells in between, a cluster of cells in the interstitial space inside the testis. And so, these Leydig cells are otherwise known as interstitial cells. Okay, so we can say that these are present in the interstitial space in the testis. That is, in between the seminiferous tubules, you can see the Leydig cells. And what is the function of these Leydig cells? These Leydig cells secrete the male androgens, isn't it? The male androgens like testosterone. The male hormone, right? The male sex hormone, the testosterone, it is secreted by the Leydig cells, which is seen in between the seminiferous tubules. That means the interstitial space, and they are basically known as interstitial cells. Okay, then what is option C or the question C? Here, the male accessory glands. And what are the male accessory glands? We know that the male accessory glands are the prostate gland, then seminal gland, and Cowper's gland, where Cowper's gland is otherwise known as bulbourethral gland, and they constitute the seminal plasma. The secretions from these glands along with the sperm constitute the seminal plasma, right? The semen, right? So, they constitute seminal plasma. The seminal plasma along with the semen form the seminal plasma along with the sperms produce the semen, constitute the semen, right. The seminal plasma is actually secreted by these glands, the prostate gland, the seminal gland and the Cowper's gland or bulbourethral gland. And the secretions from the bulbourethral gland, they also provide lubrication for the sexual intercourse. So the functions we can say it is basically having fructose, then prostaglandins and other prostaglandins, and fructose is basically they helps in the nourishment of the sperms, right. The energy that is required for the transport of the sperms, everything is actually provided by the secretions only, and along with that it helps in lubrication also. Lubrication. Okay. So let us move on to the next question. Identify the polarity of X to X prime or X dash in the diagram given below and mention how many more amino acids are expected to be added to this polypeptide chain. So this is the structure of a translational process, right? The translation is happening here, and the translation is the formation of proteins, right? The formation of proteins from an mRNA is known as translation. And we can see that the ribosome is traveling from this end to this, right. So where the translation begins is the 5' end of the mRNA. The translation will begin from the 5' and the ribosome will move from 5' to 3', right. The ribosome will move from the 5' to 3' end of the mRNA. So it has started from here only, right. So we can say that the X represents the 5' end and X dash represents the 3' end of the mRNA. Okay. And mention how many more amino acids are expected to be added to this polypeptide chain. So here you can see that the last amino acid is valine, right? And it is coding GUU. Then the next code, what is the next code here? Codon here, it is UAA. UAA. What is the speciality of UAA? UAA, UAG and UGA. These are the stop codons. Once the ribosome has reached the stop codon, it will not continue the translation. The translation will be stopped at this particular point only. So we can say that no more amino acids will be added to this polypeptide chain. Okay. Next question. Mention the codon and anticodon for alanine. Okay. So let us see the alanine here. The alanine is here, right? And the codon for alanine is GCA. Okay. So the codon for alanine. Codon is GCA. And we need to write the anticodon also, right? The anticodon which will be there in the tRNA it will be corresponding to this codon, right? So here in complementary to G, you can see C will be there, complementary to C, G, and for A, it will be U, uracil. So codon is GCA and anticodon is C, G, and U. Why are some untranslated sequences of bases seen in mRNA coding for a polypeptide? So in case of mRNA, if this is the coding region, you can see some untranslated region or UTR. Okay, UTR. And UTR, what is the purpose of UTR? It is required for the efficiency, in order to ensure the efficiency of translation. In order to ensure efficiency of translation, we require UTR, untranslated region. Okay, And where it is actually seen, where are exactly they present on mRNA? They are present on the 5' or means, that means they are present on the beginning of the the coding sequences and at the end of the coding sequence also. Okay, so we see that they are present on the beginning of the beginning of initiator codon, that is AUG. Just before AUG you can see the untranslated region here. So once this is UTR, that means here you can see AUG, the initiator codon, and after the stop codon, that means UAA or UAG or UGA, you can see the untranslated region. So on the beginning of the initiator codon and after the terminator codon, termination codon also, you can see the untranslated region, so the UTR. Okay, next question. How is Hardy-Weinberg's expression p square plus 2pq plus q square equal to 1 derived, and list any two factors that can disturb the genetic equilibrium? Okay, so we can say the second answer. We can answer the second part first. So here we can say the genetic equilibrium is actually affected by certain factors like gene migration or gene flow, then recombination, mutation, then genetic drift, etc. Okay, genetic drift, etc. So let us go to the first part of the question. How is Hardy-Weinberg equilibrium, that expression, is derived? So we know that the sum total of all allelic frequencies in this population is 1, right? So that means P plus Q is equal to 1, where P represents the allelic frequency of A gene. Suppose P represents the allelic frequency of A gene, okay, and the allelic frequency of small a gene is represented by Q, suppose. Okay, and we know that sum total of all alleles in a population, we know that, right? Sum total of all alleles in a population is 1. That is P plus Q is equal to 1. Okay, so if you are going for a cross like this, a heterozygous cross here, capital A small a into capital A small a, we will get 4 kinds of offsprings, right? Where it will be capital A, capital A small a, capital A small a, and small a, small a. Okay, they will produce 2 kinds of gametes, and each of them when fertilized they will produce 4 types of progenies. Okay, so let us see the allelic frequency. The allelic frequency of capital A will be equal to capital A, here it is P into P is equal to P square. Right. Then allelic frequency of small a will be equal to Q into Q, that is Q square. Right. Then let us see. Let me write here allelic frequency of capital A small a. See, capital A small a, that means capital A, here P into Q, right? And how many is there? 1, 2. 2 is there, right? So 2 PQ, 2 into PQ, right? And we know that the sum total, all of the progeny, sum total of all this progeny is that total allelic frequency. We can write it like this: total frequency, allelic frequency is equal to, see, P square plus q square plus 2pq. Right, p square plus q square plus 2pq. Right, and we know the mathematical expression where p plus q the whole square is equal to p square plus q square plus 2pq. So we can write it like this. P square plus q square plus 2pq is equal to p plus q the whole square, where p plus q is equal to 1. So p plus q the whole square is equal to 1 square, that is 1. So finally we will get p square plus q square plus 2pq is equal to 1 only. Okay. So this is how we get the Hardy-Weinberg's expression. Next question. Highlight the structural importance of an antibody molecule with a diagram. Name the 4 types of antibodies found to give a humoral immune response, mentioning the functions of 2 of them you have studied. Okay, and it is having an R question also. Let us see that too. Explain the life cycle of Plasmodium starting from its entry in the body of female Anopheles till the completion of its life cycle in humans. Okay, and B, explain the cause of periodic recurrence of chill and high fever during malarial attack in humans. Okay, so let us see the first question. Here, highlight the structural importance of an antibody molecule. So we need to explain the structure of an antibody molecule with a neat and labeled diagram, and name the 4 types of antibodies which are supposed to give a humoral immune response. We know that humoral immune response are the response offered by the B lymphocytes, right? So we need to specify the types of antibodies there and mentioning the functions of any 2 of them that we have studied. Okay, so let us see the answer of that particular question. So we know that antibody, it is having a light chain. It is having a Y-shaped structure. Somewhat a Y-shaped structure it is having. Okay, here it will be having 2 heavy chains like this, and 2 light chains also. 2 light chains also. Okay, it will be having 2 heavy chains and 2 light chains, where the light chain is connected to the heavy chain by means of disulfide bond. Disulfide bond. So we know that an antibody, it is having a Y-shaped structure. It is composed of 2 heavy chains. We can say that it is having 2 heavy chains and 2 light chains. Okay, 2 heavy chains and 2 light chains they are having, and the light chains are connected to the heavy chain by means of a disulfide bond. A disulfide bond, we can say. Okay, it is connected. The light chain is connected to the heavy chain by means of a disulfide bond, and similarly the heavy chains, both the heavy chains are connected together by means of disulfide bonds. Here also you can see disulfide bonds. This is the hinge region of the heavy chain. So the light chains are also interconnected by means of disulfide bond. Similarly, the heavy chains and the light chains are connected to the heavy chains by means of disulfide bond. Clear? Okay, then we can say that both the light chain and the heavy chains are having an antigen binding site here. See, here they will be having an antigen binding site. Okay, here you can see they will be having an antigen binding site. So we can say that an antibody will be having 2 antigen binding site. The site where the antigens are bound, okay, for cleavage. Okay, they are destructed. These antibodies are attached to the antigens by means of through this antigen binding sites. Okay, so they will be having 2 antigen binding sites, and here the stalk-like region here, over here, the stalk-like region is known as the FC region. FC region, and the tip here, the variable region, both the heavy chains and the light chains will be having a variable chain over the top, and they are represented like this. So here you can say that the variable region of the light chain is represented by VL, and the variable region of the heavy chain is represented by VH. So they will be having 2 variable region each on the light and heavy chains. Okay, so this is the basic structure of an antibody. Okay, so they are having a Y-shaped structure where the light chains and heavy chains are there and they are interconnected. The heavy chains are interconnected by means of disulfide bonds. Similarly, the light chains are connected to the heavy chains by means of a disulfide bond. And here this is the hinge region, and see here in the tip, on the lower end you can see an FC region there, and the upper end they are having a variable region where the light chains will be having VL region and the heavy chains are having VH region. These are the variable regions. Okay, and we can represent an antibody like this H2L2 because it is having two heavy chains and two light chains. Okay, that was the first part of the question. And what was the second part? See, name the 4 types of antibodies found to give a humoral immune response, mentioning the functions of the 2 of them you have studied. So, we know that the antibodies are otherwise known as immunoglobulins. Okay, because they are providing us with immunity and they belong to a class of glycoproteins known as globulins. Okay, they are belonging to a class of glycoproteins known as globulins, and they are known as, these antibodies are known as immunoglobulins, and the antibodies are of different types, isn't it? We can say IgG is there, then IgA, then IgM, IgE, that is epsilon, and IgD. 5 types of antibodies are there. Okay, and let us see the functions of any 2 of them. See here IgG, we can say that IgG is the antibody which crosses the placenta. It is capable of crossing the placenta. It is the only antibody which is capable of crossing the placental barrier. So, they are basically providing a baby with a passive immunity. Okay, they are providing a passive immunity to the baby. Clear? Then, IgA, it is present in breast milk, mucus and blood, etc. So, they are basically provided by lactational mothers to their child. Okay, then IgE, it deals with allergic reactions. It deals with allergens and they are responsible for hypersensitivity or allergy, you can say. Okay. Okay, and IgD helps in the differentiation, D for differentiation. So, we can say they helps in the differentiation of B lymphocytes. They help in the differentiation of B lymphocytes. Okay, and the IgM, they are the largest in size. Where the largest in number is the IgG, IgM is largest in size, and they are the first to come. Okay, so these are something about the various immunoglobulins. Okay, so the second part, our question was, explain the life cycle of Plasmodium starting from its entry in the body of female Anopheles till the completion of its life cycle in humans. Okay, and explain the cause of periodic recurrence of chill and high fever during malarial attack in human. So, we need to explain the life cycle of Plasmodium, right? So, we know that Plasmodium is basically digenetic in nature. It is digenetic in nature. What do you mean by digenetic? Digenetic, because it needs 2 hosts to complete its life cycle. It needs 2 hosts, isn't it? One is human and the other is mosquito. So, basically they require 2 hosts to complete its life cycle. Okay, so when the mosquito is like an infected mosquito is injecting or it is attacking or biting a person, a healthy person, we know that the mosquito, the saliva of mosquito will be having, it will be having sporozoites, okay, sporozoites of the plasmodium. Okay, sporocyte is a larval form. Sporozoites of the plasmodium is entering. It is injected into a human body. Okay, it will enter inside a human body. And there inside the human body, we can show it like this. Inside the human body, these sporozoites will undergo like a development. It will undergo gradual development to form certain stages known as merozoites. Okay, then they will produce trophozoites and schizont. So these are the asexual life cycle of this sporozoite, and it takes place inside the liver. Everything is happening inside the liver. Okay, inside the liver only. Okay, and what happens is that it continues finally rupturing the hepatocyte. That means the liver cells which they have attacked. So once if they are entering inside a liver cell, they will produce numerous trophozoites and schizonts there, and once they are producing sufficient amount of schizonts or the other larval forms, what happens is that they will rupture, releasing these to the outside, and they can attack another hepatocytes or the liver cells. Okay, and in some cases they will go and enter inside the RBC. Where inside the RBC, what happens is that they will produce the gametocytes. Okay, and again these gametocytes are taken up by the mosquito, and inside the mosquito, what happens there? Inside the mosquito, the gametocytes will undergo fertilization. The male and female gametocytes will undergo fertilization and development, zygote formation. Okay, development of zygote, the sexual cycle, and they are from the mosquito, the sporozoite, they will produce sporozoites, and the sporozoites are injected into the human, and the life cycle continues. Okay, the life cycle continues here. So this is the life cycle of Plasmodium. It is digenetic in nature, right? Then what was the second part of the question? Explain the cause of periodic recurrence of chill and high fever during malarial attack in humans. So this occurs when the RBCs rupture. So I said that the RBCs rupture, releasing the gametocytes, right? So the rupture and release of RBC results in the release of a toxin known as hemozoin. It is a toxin. Okay, so it is because of the release of this hemozoin, periodic recurrence of chill and high fever occur during a malarial attack in humans. Clear? Okay, so let us move on to the next question. Question number 29 and 30 are case-based questions, and each question has subparts with internal choice in one part, and each carries 4 marks. Okay, so let us see the 29th question here. Study the pedigree chart given below and answer the questions that follow. Okay, so we are given a pedigree chart here, right? Let us observe it. So here you can see that a carrier male is married to a carrier female, and they are giving birth to an affected male. Okay, and similarly you can see a carrier male is again here married to a carrier female, and they are giving birth to a affected male and a normal female. Okay, so we can say that a male, he'll be having an X chromosome and a Y chromosome, right? Here female, she'll be having an X chromosome, 2 X chromosomes, right? Here also you can say here an X and a Y here, 2 X chromosomes. See, let us observe here. In this pattern of inheritance, when 2 carriers are married, the male is affected. Here also the 2 carriers are married and the male is affected, but the female is not affected here. So definitely we can say that this might be linked to the X chromosome, right, where males will be having only one X chromosome, and there is a possibility that if the X chromosome is affected, then there is no replacement for that, then definitely that person will be affected. So we can see that it is something related to the X chromosome, and see that they are expressing the disease only if both are carriers, right? And the possibility is that if they are, both the parents are carriers, there is a possibility that they might produce an affected person. Similarly, here also we can say the same. Okay, then if we are considering the second generation, here an affected male is married to a normal female, and see the offsprings, their progeny, see one affected female is there, then one affected male is there, and one normal child is also there. So we can say that they are expressing, all the progenies are expressing this character only if both of them are what? Having a defective gene. Okay, see here both of them are having defective gene, and the possibility of the progenies having the disease was more, right? So we can say that this disease is something related to X-linked, and along with that we can say that recessive trait, X-linked recessive trait. Okay, because definitely see both of them are carriers and the male is getting affected here. Here also both of them are carriers and the male is getting affected, but the female is not getting affected. The possibility is that one of the normal X chromosome from the mother might have substituted the defective X chromosome. Here there is nothing for substituting there, so both the males are getting affected. Okay, so let us see the first question. On the basis of the inheritance pattern exhibited in this pedigree chart, what conclusion can you draw about the pattern of inheritance? We already said that, right? It is an X-linked recessive trait. Okay, then if the female is homozygous for the affected trait in this pedigree chart, then what percentage of her sons will be affected? So what is said here? If the female is homozygous, suppose we can say, we said that it is an X-linked disease, right? So the defective X chromosome, we can represent it like this. Okay, and a normal X chromosome we can represent like this. Okay, and what is said here? The female is homozygous for the affected trait. That means both the X chromosomes are defective. Okay, in this pedigree chart, then what percentage of her sons will be affected? Let us see. If the female is affected, that means it is homozygous, means both the X chromosomes are having the defective gene. Okay, so we can say that she will be producing two kinds of ovum, and all the ovum means she will be producing ovum which will be having all the defective X chromosomes. Okay, and if she is married to a normal male, see the male will be producing two kinds of sperms. One will be having a normal X chromosome and the other will be having a normal Y chromosome. And see the number of sons she will produce here, the possibility of the sons that she will be producing here. See here, all the sons will be having the defective X chromosome, because the mother doesn't have a normal X chromosome, right? So 100% of her sons will be having a defective X chromosome. That means 100% of her sons will be affected. Okay, clear? Then give the genotype of the offsprings 1, 2, 3 and 4 in the third generation. Let us see the genotype of 1, 2, 3 and 4 of the third generation. See here the male is carrier. It is shown that he is carrier, but sometimes he will be affected also. It is said that he is having one X chromosome, defective X chromosome. So we can represent it like this. Either he will be carrier or sometimes he will be normal. We don't know the exact status of him. Okay, so sometimes it will be XY, or there is a possibility that he will be having a defective X and a normal Y, XY or X-Y. Okay, and here second, the second individual here, she is affected, right? She is affected. That means she is having both the defective X chromosomes. Okay, then here the third individual, here he will be having a defective X chromosome and a normal Y chromosome because he is affected. Okay, and the fourth one here, she is not affected. That means she is having two normal X chromosome. Okay, clear, right? Then next, in this type of inheritance pattern, out of male and female, which one has less probability of receiving the trait from the parents, and give a reason. So we can say that the probability of receiving the trait from their parents is lesser in case of females, because females get affected only if both of her parents are affected. Either her parent, the mother should be carrier and the father should be affected. Only if such a condition happens, there the female will be receiving both X chromosomes, so that she will be receiving the trait from the parent. Okay, so the probability of receiving the recessive trait is lesser in case of females. She will receive a defective trait only if both the parents are affected. She is having a carrier. She is born to a carrier mother and an affected father. Okay, so the probability is lesser in case of females. Okay, so let us move to the next question. The data below shows the concentration of nicotine smoked by a smoker taking 10 puffs per minute. Okay, so see here it is the concentration of nicotine in blood per milligram per centimeter cube. Okay, we are considering the concentration per centimeter cube of blood. Okay, milligram per centimeter cube of blood. Okay, then what is the question here? With reference to the above graph, explain the concentration of nicotine in blood at 10 minutes. At the 10th minute, let us see here, as the time is increasing, you can see he is taking 10 puffs per minute, right? So, as the time is increasing, you can see at the 10th minute, at the 10th minute it is maximum. Okay, so what can we say here? The concentration of nicotine will be maximum at the 10th minute. Okay, and how much it is? It is 45 milligram per centimeter cube. So, we can say the concentration will be maximum at 10th minute, at 10 minutes. Okay, and we can say that the concentration is 45 milligram per centimeter cube. 45 milligram per centimeter cube of blood. Okay, and how will this affect the concentration of carbon monoxide and heme bound oxygen at 10 minutes? What happens there? Since the concentration of nicotine is more there, that means the carbon monoxide is more in concentration there. The affection, the attachment or the affinity towards, we know that the affinity of hemoglobin towards carbon monoxide is more than that of oxygen. Okay, so at this particular stage, we can say that the concentration of carbon monoxide in the blood increases. Okay, concentration of carbon monoxide increases where the concentration of heme bound oxygen decreases. Clear? Then how does cigarette smoking result in high blood pressure and increase in heart rate? Okay, so cigarette smoking, that means the nicotine increases, or it basically stimulates the adrenal glands. And they result in the increased concentration of adrenaline, increased production of adrenaline and noradrenaline, where Both of them are emergency hormones, right? They increases the blood pressure. These adrenaline and noradrenaline increases the blood pressure and the heart rate. Clear? Then, how does cigarette smoking result in lung cancer and emphysema? We can say that in case of lung cancer, there are certain chemical carcinogens present in cigarette smoke, right? The chemical carcinogens in cigarette smoke result in lung cancer, where in case of emphysema, we can say that the nicotine, the cigarette smoke, basically it irritates the air passages or air pathways. The bronchioles, the alveoli, etc. The air pathways are irritated by the cigarette smoke and nicotine. And what happens there? Mucus secretion increases, right? Mucus secretion increases, accumulation of mucus increases. It results in cough, right? It results in cough. Then what happens? It results in the reduction of surface area. Reduction of surface area where air exchange takes place. The elasticity, it will be lost. Okay, it will result in the reduction of elasticity, and finally it results in emphysema and difficulty in breathing. Okay, then next question. Answer the questions from 31 to 33. Each carries 3 score. Okay, so trace the events from copulation to zygote formation in a human female, or trace the development of a megaspore mother cell to the formation of mature embryo sac in a flowering plant. And this is a question from the botany part. Let us discuss the zoology part of this question. Trace the events from copulation to zygote formation in a human female. Okay, so let us see the answer here. We know that females are producing that means the mother, females will be producing what? Oocyte or secondary oocyte. You can say secondary oocyte. At the time of ovulation, they will release secondary oocyte, and males, they will release the sperms. Okay, the sperms. And the sperms are ejaculated. They are ejaculated and are inserted or introduced into the vagina by means of insemination. The process is known as insemination. The release of sperms into the vagina of a female is known as insemination. Right. And from the vagina, it travels down to up into the cervix, and it passes the uterus. Finally, it will reach the ampulla region of the fallopian tube. Ampulla region of fallopian tube or the oviduct. Okay, or the oviduct. There, in the ampullary region, the fertilization takes place. Okay, so this results in the fertilization, where the male and the female gamete will fuse together to form the zygote. Okay, and during fertilization, what happens there? During fertilization, we can say that here it is the fallopian tube. During fertilization, the sperm will touch the ovum. Here you can say that the ovum will be having, that means the secondary oocyte will be having a covering known as the zona pellucida. Zona pellucida. Okay, so the sperms will reach the zona pellucida, they will touch the zona pellucida, and the sperm head is having acrosome, right? Acrosomal proteins will be released there. It helps in the penetration, okay, penetration of zona pellucida, penetration of zona pellucida. Okay, zona pellucida happens there, and what happens then? It results in certain changes in such a way that it prevents polyspermy. That means no more sperms are permitted to enter inside the ovum. Okay, then once the sperm has touched the secondary oocyte, what happens there? It completes second meiosis, right? It completes second meiosis. Second meiosis, and you can say that the second meiosis unequal in action, and it results in the formation of an ovum or an ooted. You can say an ooted and a polar body. Polar body. A polar body will be formed. Okay, and then what happens there? The sperm has penetrated, right? The sperm nucleus and the ovum nucleus, the nucleus of the ovum will fuse together. The sperm nucleus and the ovum, nucleus of the ovum will fuse together to form a diploid zygote. A diploid zygote. The haploid sperm and haploid ovum will fuse together to form a diploid zygote. So this is the event. These are the events that occur during fertilization. Clear? Then, next question. Observe the segment of mRNA given below. See, you can see an mRNA here and it is having exons and introns, right? So, this is not a perfect mRNA. We can call it hnRNA. hnRNA. Heterogeneous nuclear RNA. Okay, so what is the question here? Explain and illustrate the steps involved to make fully processed hnRNA. So these are the one we saw was an hnRNA, right? And we need a fully processed hnRNA. That means we need a fully processed mRNA. And what are the steps involved there? See, what are the steps involved there? Let us see. First what happens is that a capping occurs. A capping occurs. Capping at the 5' end. At the 5' end, what happens? Methyl guanosine triphosphate will be added. Methyl guanosine triphosphate will be added. So a cap occurs here. A cap is added at the 5' end. Here you can see a cap region. Okay, a cap will be added at the 5' end and the cap is of methyl guanosine triphosphate. Then what happens? Tailing occurs. Tailing occurs. Here at the 3' end, 200 to 300 adenylate residues are added. Adenylate residues are added. See, here in the 3' end, numerous adenylate residues will be added. This is the polyadenyl A tail. A poly A tail will be formed. So, capping occurs, tailing occurs, and finally what happens then? Splicing occurs by means of spliceosomes. Spliceosomes will be formed. Splicing results in the elimination. It results in the elimination of introns. Introns will be removed by means of splicing. And finally, we will get a mature or processed mRNA. Clear? Then, what is the next question? Gene encoding RNA polymerase 1 and 3 have been affected by mutation in a cell. Explain its impact on the synthesis of polypeptide, stating reasons. So, RNA polymerase 1, it is involved in the transcription of what? RNA polymerase 1 is involved in the transcription of rRNA. That means ribosomal RNA is transcribed. It is actually made, the ribosomal rRNA is made by means of the RNA polymerase 1. And they are basically the protein factories, right? The ribosomes are acting like the protein factories. They are translating the mRNA. The translation of mRNA is done with the help of the ribosomes, right? The ribosomes. And if the ribosomal RNA is not formed, what happens there? What happens there? No translation will happen. Okay. Then, similarly, RNA polymerase 3. RNA polymerase 3 is involved in the transcription of tRNA, where tRNA is basically carrying the amino acids that are needed for the translation of mRNA. They are basically carrying the amino acids, right? They are having a they are basically carrying the amino acids that are necessary for translation of the mRNA. So, since both RNA polymer is 1 and 3 are absent or they are defective, that means that the translation of mRNA will not occur. Translation of mRNA will not occur. Okay, it will not occur. Then, next, study the schematic representation of the genes involved in the lac operon given below and answer the questions that follow. Let us see that image here. See, you can see a lac operon here, right? This is the lac operon. Lac operon. It is involved in lactic acid metabolism. And see, the active site of enzyme permease present in the cell membrane of a bacterium has been blocked by an inhibitor. How will it affect the lac operon? And the enzyme permease, it is basically helping in the entry. It permits the entry of lactose into the cell, right? Permease basically permits the entry of lactose into the cell. Okay, and what is the condition here? The active site of enzyme permease present in the cell membrane has been blocked. So that permease, the activity of the permease will not happen there. So what happens? What it results? It results in the hindrance. Okay, it results in the hindrance of the entry. So basically we can say that what will happen there? Lactose will not enter inside the cell. Permease is basically permitting the entry of lactose into the cell. It is hindered. What happens because of that? Lactose cannot enter into the cell. So, lactose cannot enter into the cell as a result of this. Clear? Then, the protein produced by the I gene. See, the protein produced by the I gene has become abnormal. We know that the protein produced by the I gene is the repressor protein. And the repressor protein will go and bind to the operator region in such a way that the operon will not function. Right? It will not function. When the repressor protein is bound to lactose, which acts as an inducer, what happens that it removes from this operator region so that this can be translated. This is the normal condition of lac operon. But here, the repressor protein, it is abnormal due to some unknown reason. Explain its impact on lactose metabolism. What happens if the repressor protein is defective? It cannot bind to the operator region. Okay, so continuous operation occurs, isn't it? Continuously, the lac operon will be working. The lac operon will be working continuously. Okay. It will not be switched off. It is switched on continuously. And see the last part of the question. If the nutrient medium for the bacteria contains only galactose, will operon be expressed? Justify your answer. Galactose, it is not an inducer. Only the lactose act as an inducer, right? Lactose or allolactose act as an inducer for this operon. Galactose is basically the product of lac operon. It is the product of lac operon. Isn't it? It is not an inducer. Inducer is lactose. So, what is your answer? Will the operon be expressed? No, the operon will be expressed only in the presence of lactose. So that's all about the questions from the zoology part in the sample paper. Hope you all understood the answer. Thank you.
Hi my dear students, welcome to the session. Today we are going to discuss about a sample question paper of 2022-23 of CBSE. Right, and I'm going to discuss about the botany part of this question paper. Right. you know that there will be certain instructions that you have to follow here. All questions are compulsory questions, okay, and you'll be having 5 different sessions, okay, that is A to E sessions, and each of the questions will be having certain OR questions in them, okay, individual options will be there, okay. So, first part, that answer the questions from question number 1 to 16 and each carries 1 mark, okay. So, the first session will be carrying it will be having questions which will carry 1 mark each. Okay, so let's see the questions. So, first one, which of the following amino acid residues will constitute the histone core? Okay, so you know about what is a histone. Okay, so what do you mean by a histone? Histone is actually a protein, right? So, in that protein, okay, the long DNA is being wrapped around, right? So, the histone proteins will be there and it will be forming a histone octamer, and in which the long DNA will be wrapped around, right? That you know, that is the nucleosome. That structure is called as the nucleosome, right? So, the question is that the amino acid residues which will constitute this histone core. Okay, so which amino acid is responsible for the formation of these histone proteins or the histone core? Okay, and the options are lysine and arginine, right? Asparagine and arginine, glutamine and lysine, asparagine and glutamine. Okay, and from this we can choose that lysine and arginine. Okay, so these lysine and arginine are the 2 amino acids which will constitute the histone core. Okay, so that will be the answer that you have to keep in mind this. Then, evolutionary convergence is development of A. Okay, and here you can see a question from evolution, right? So, evolutionary convergence, right? So, we know that convergent evolution and divergent evolution, right? So, in case of divergent evolution, what will happen? That is, from a common ancestry, okay, from a common ancestry, the evolution is being happened and it will be forming different variations, okay, variety of organisms will be forming from a common ancestor. Okay, so that is a divergent evolution. But in case of convergence or convergent evolution, what will happen? Different organisms will be there, divergent organisms will be there, and they will be forming a common character. Okay, it will be after the evolution, they will be forming a common organ. Okay, and which will be forming the same functions, okay, which will forms the same follow the same functions. Okay, so from different organism, a single organism is being formed. Okay, that is the evolution is converging, that is convergent evolution, right? So the question is, evolutionary convergence is a development of, okay, let's see the options. Common set of functions in groups of different ancestry, then dissimilar set of functions in closely related groups, then common set of structures in closely related groups, dissimilar set of functions in unrelated groups. Which will be the answer? Right, that is a common set of, right, a common set of functions, okay, in groups, different groups, a common set of function of different ancestry, okay, different ancestry, they will be having a common set of function, right? So that is evolutionary convergence. Okay, it is a development of a common set of functions in a group of different ancestry. From group of different ancestry, it will be forming a common set of function in single organism, right? Then here you can see a column, that is which of the following water samples. You can see different water samples A, B, C and D. Okay, so which of the following water samples in the table given below will have a higher concentration of organic matter? High concentration of organic matter that you can find in which of these water samples? That is a question. Okay, so water sample A will be having a level of pollution high and its value of BOD. So what do you mean by BOD? That is the biological oxygen demand, right? And this is the oxygen demand by which the organism, the microorganisms that is present in the water body, it will require certain amount of oxygen for the disintegration or for the decomposition of the organic matter that is present in the water body. Okay, so that is the BOD, or the biological oxygen demand of the water body. Right, so here you can see the level of pollution and the value of BOD in each of the water samples is given in this table. Right, so in water sample A, you can see a high level of pollution. Okay, high level of pollution, so high pollutants will be there and its value of BOD is very high. Similarly, right, and in water sample B, level of pollution is very low, but the value of BOD is again low, and in water sample C, you can see level of pollution is low and the BOD value is very high, and in D, level of pollution is high but the BOD value is low. Okay, and you can remember one thing, that if an organic matter, high amount of organic matter that is present in the water body, okay, so that it requires more oxygen, okay, by the bacterial organism or the bacteria to decompose these high amount of organic pollutants. Okay, then only they can use the, or they can convert that organic substances into simple forms. Right, so for that, in a water body will be having a pollution level very high pollution, that indicates, okay, the oxygen demand in that water body is very high. Okay, so that it requires more amount of oxygen to decompose the high amount of pollutants that is present in the water body, right? So the BOD, biological oxygen demand, is also very high in a high polluted water body. Okay, so in this water sample A will be having a high level of pollution. Right, so that means it requires more oxygen to decompose the organic matter present in the water body. So the BOD level is also will be very high, right? So this will be the correct option. Okay, so the water sample, that is the water sample A in the table, that will show a high organic concentration of the organic matter, right? So that will be the answer. The figure below shows the structure of a plasmid. Okay, so that you well know about the structure of a plasmid, that you know, that is PBr 322. Okay, it will be having an ORI, that is origin of replication, the proteins for replication ROP, and two antibiotic resistant sites, AMPR and TETR, ampicillin and tetracycline, and here you can see a BAMH1 site, that is a restriction site, right? So a foreign DNA was ligated at this BAMH1 site. Okay, here you can see the ligation of a foreign DNA. The transformants were then grown in a medium containing antibiotics, tetracycline and ampicillin. So choose the correct observation for the growth of the bacterial colonies from the given table. Okay, you can see a table here, that is a medium with tetracycline, okay, and a medium with the ampicillin. Okay, whether the bacterial cell is grown in this tetracycline medium or in this ampicillin medium, that you have to find here. Okay, so here you can see the gene of interest was ligated in this site, in BAMH1 site. Okay, so you know that when the genes are inserted in this plasmid, okay, so then the plasmid is being recombinant. Okay, then in some cases there is non-recombinants will also be formed. Okay, that the gene of interest does not combine with this site, okay, or the region of this plasmid. Okay, so that is a plasmid which is a non-recombinant one, and the gene of interest is being ligated, then it is called as a recombinant one. Okay, then it will be ligated into the, or it will be inserted into the bacterial cell. Okay, so transformants are of two kinds, that you know, that is recombinant transformants. Okay, recombinant transformants and non-recombinant ones. Okay, non-recombinant transformants. That means a transformant or the bacterial cell which will be having this foreign gene inserted in it. Okay, that is a recombinant transformant, or the transformant or the bacterial cell which do not have the this foreign gene ligated in it. Okay, that is a non-recombinant transformant. Okay, so what is the condition here? That is these transformants, when they are grown in a medium which is containing antibiotics, that is tetracycline and ampicillin. Okay, they are grown in this tetracycline and ampicillin medium, and you can see what is their growth condition, that you have to find here. Okay, so in this transformants, when they are grown in this ampicillin medium, what will happen actually? In ampicillin medium, if the transformants are grown, what will happen? They have the ampicillin resistance because there is no hindrance, okay, of this resistance condition. Okay, so they will grow in this ampicillin medium. But in this tetracycline region, okay, this TETR resistance site, in this site what will happen? When this foreign gene is getting inserted, they lose their tetracycline resistance. So when they are recombinant, okay, when they have this gene of interest, they are, what will happen? They lose their tetracycline resistance. So they will no longer survive in the tetracycline medium. Okay, so let us see that in tetracycline medium, what will happen? You cannot find any growth of the bacterial colonies. Okay, no growth is being observed. And in the ampicillin medium, what will happen? They are very much grown. Okay, so that is, they have the ampicillin resistance, but they do not have this tetracycline resistance. Right, so that is the answer here. Okay, then next question. Swati was growing a bacterial colony in a culture flask under ideal laboratory conditions where the resources are replenished. Then, which of the following equations will represents the growth in this case? Okay, and here this equations, for this equation where population size is n. Okay, n is the population size. Population size, that is n, and birth rate is b, death rate is d, and the unit time, okay, for the growth is t, and carrying capacity of the population is k. Right, that you know. So, in population growth, you know that there will be two types of growth patterns. Okay, a logistic growth as well as an exponential growth. Right. So, in this condition, what happens is that here a bacterial colony in a culture flask is growing under ideal laboratory condition where the resources are replenished. That is the clue. Okay, the resources are getting replenished. Okay, so for resources they are getting replenished, what type of growth we can observe there? That is an exponential growth pattern that you can see there. Okay, exponential growth pattern, that is a geometric growth pattern. For a geometric or exponential growth pattern, the curve will be, what will be the shape of the curve? It will be a J-shaped curve that you can observe in exponential growth pattern or geometric growth pattern. Okay, a J-shaped curve. A J-shaped curve you can observe, that is for geometric growth pattern. Right. Geometric or exponential growth pattern. That is, the resources are replenished. Okay, so when we are used up, they are again and again, they will get replenished. So you can observe a geometric growth pattern. For a geometric growth pattern or for getting a J-shaped curve, the equation of the population will be, what will be the equation? That is, the dn by dt is equal to Rn. Right. So n is the population size, t is the time, unit time, and n is the size of the population, and here R, what will be the R? That is the intrinsic rate of natural increase. R is intrinsic rate of natural increase. So, this will be the equation for this population. Okay, this bacterial colony, which will show a J-shaped curve for a geometric pattern of growth. Right. So, dn by dt is equal to Rn is the equation for this population. Then, C. anemone gets attached to the surface of the hermit crab. The kind of population interaction exhibited in this case is, okay, here you can see the association of C. anemone and the hermit crab. Okay, and here you can see what kind of interaction, that is, you can see here amensalism, commensalism, mutualism and parasitism. Right. So, here you can see that in this hermit crab, in this situation, hermit crab actually provides food for the C. anemone. Okay, they will nourishing the C. anemone. Okay, and in return, they are not giving anything, but one of that species will not getting not harmed or neither they will be getting harmed. Okay, so here one organism is benefited and the other one is not harmed at all. Okay, they are not getting any benefits and they are not harmed, right? So this is a type of what kind of interaction you can see? Yes, that is commensalism. Okay, in parasitism you can see one of the organism will be depending on a host organism. With harming the host, it will taking nutrition or nourishment from that host, right? That is parasitism. In mutualism, you can see that what is being happened, both the organism is being benefited by means of mutualistic relationship, right? Then in amensalism, what will happen? One of the organism is completely harmed and the other one is nor or neither they will be getting harmed. Okay, so that is amensalism. But here, one of the organism is getting benefit. Okay, hermit crab will provide food for the sea anemone. Okay, so you can choose commensalism here, right? So the answer is commensalism. Then, next question. Which of the following food chains is the major conduit? Okay, major conduit for energy flow in terrestrial and aquatic ecosystems respectively. Okay, and if you you are studying about the ecosystem and all, okay, in aquatic as well as terrestrial ecosystem, when you are studied that, then what is the thing that you have to keep in mind? That is in terrestrial ecosystem, in land ecosystem, you can see that detritus food chain is, it is the main conduit of energy flow. Okay, detritus food chain is the main conduit of energy flow in terrestrial ecosystem. But in aquatic ecosystem, you can see that grazing food chain is the main conduit or energy flow. Okay, so you can choose in terrestrial ecosystem, the detritus ecosystem. In terrestrial ecosystem, detritus food chain is the main conduit for energy flow. Okay, so more amount of energy is transferred by means of this detritus food chain. Okay, but in aquatic ecosystem, you can choose that is grazing food chain. Okay, grazing food chain is the main conduit of energy flow. Okay, more amount of energy is being transferred through the grazing food chain. Okay, so you can choose option C, that is terrestrial ecosystem is detritus and aquatic ecosystem is grazing type of food chain. Okay, that they will be the main conduit for energy flow, right? Then, which of the following is an example of ex situ conservation? Okay, you studied well about ex situ as well as in situ conservation, right? So, in situ conservation, what will happen? That is, an organism is being conserved in its natural habitat. Okay, so without disturbing its natural routines, right? So, that is in situ conservation. But in ex situ conservation, what will happen? An organism is taken from its natural habitat into an artificial conservation area, right? So, that is an ex situ conservation. And here you can see in secret groves, okay, you cannot see any kind of ex situ conservation there, okay. That is the organism, if it is plants or animals, okay, whether they are always present in their natural habitats without disturbing them, right? So, secret groves are in situ method. National parks, okay, here also you can see we are not disturbing that organism that is present in their natural habitat in national parks, right? Then in biosphere reserves or, okay, so these two is a kind of an in situ method of conservation, right? In biosphere reserves also, the human activities are very much restricted because they weren't able to distract the activities of that organisms in that area, right? So, that biosphere. Then seed banks, you can see in seed banks, we can take the seeds of economically beneficial or important plants. Okay, so we can collect that from its natural habitat and conserve it in a different area. Okay, in a laboratory condition, you can conserve that seeds. Okay, important seeds. So, that is seed bank is a type of an ex situ conservation method, right? So, you can choose seed bank. Then, next part, that is question number 13 to 16, which is consist of two statements. Okay, assertion and a reason. Two statements will be there. Answer these questions selecting the appropriate options given below. You can choose options like this A, B, C and D. Like if both assertion and reason are true and reason is the correct explanation of assertion. Okay, you can choose option A. Then if both A and R are true but R is not the correct explanation of A, then you can choose B. Right. Then if A is true but R is false, then you can choose C. Okay, if the assertion is false but the reason is true, then you can choose option D as the answer. Okay, so let's see the questions. Question number 13. The assertion given is apomictic embryos are genetically identical to their parent plant. Right. So, what do you mean by apomixis? Right. That is the formation of seed without fertilization. Okay, so production of seeds without fertilization is termed as apomixis, right? So here the seeds are producing without fertilization. So the seeds are the exact copies of their parent, right? So the first statement, that is assertion, is true, right? In that sense, so apomictic embryos are genetically identical. They are identical to their parent plant. Obviously, so this is a correct statement. And the reason, apomixis is the production of seeds without fertilization, right? So this is also true. So you can choose option A as the correct option, right? So assertion and reason are true, and reason is the correct explanation of the assertion, right? Then next one, again assertion is functional ADA-cDNA genes must be inserted in the lymphocytes at the early embryonic stage. So what do you mean by ADA? That is adenosine deaminase. Okay, so in ADA deficiency conditions, adenosine deaminase deficiency. Okay, so in that conditions, we are going for gene therapy. So that deficient ADA gene will be replaced by a functional ADA gene. Okay, in that cases, in the early embryonic stage. So we are collecting the functional genes or functional genes of this aDNA from the bone marrow, and we are inserting in the early embryonic stages in that functional gene. Okay, then only we can cure this ADA deficiency permanently. For the permanent cure, we can collect that from the bone marrow and insert it in an early embryonic stage for the permanent cure of this problem. Okay, so gene therapy is done at the early stages, okay, early embryonic stages. So in ADA-cDNA, that is functional ADA-cDNA, that is a functional ADA gene, okay, that must be inserted in the lymphocytes of an patient or an organism at the early embryonic stage. So assertion is a right thing. Right. And the reason, cells in the embryonic stage are mortal, differentiated and easy to manipulate. So this is not at all true. Okay, so the statement R is a false statement. So you can choose option C as the correct option here, because assertion is true but the reason is false. Okay, this and the reason is not the correct explanation for this assertion. Okay, for the correct explanation is what? For the permanent cure of this problem, we can collect the functional ADA genes from bone marrow and insert it in during the early embryonic stages for the permanent cure. Okay, so that will be the reason, right? So here you can choose option C as the correct one, right? Then, given below is an age pyramid of population in one of the states in India as per 2011 census. So it depicts the male population on the left hand side and the female population on the right hand side, newborns towards the base and gradually increasing age groups as we moving from base to the top. That is the old age peoples are representing the top of the pyramid. So with the oldest population at the top. Right. So study this pyramid, age pyramid, and comment upon the appropriateness of the assertion and the reason. Here also a assertion reason type question. Okay, so let's see the pyramid first. So this is the age pyramid given, and here you can see in the left side male population and the right side female population is representing, and old age peoples are representing at the top of the pyramid, newborns will be at the base of the pyramid. Okay, so this is the first levels. Okay, so pre-reproductive, reproductive and post-reproductive ages. Okay, so that is being representing in this pyramid, age pyramid, and the assertion is it is a stable population. Obviously right. So this is a stable population. We cannot expect any decreases or increases in this population, right? So this will be a stable population. And the reason, what is the reason? The pre-reproductive and reproductive individuals are almost in equal numbers, and the post-reproductive individuals are relatively fewer here, right? So the pre-reproductive and reproductive individuals. Okay, so the pre-reproductive and reproductive individuals are somewhat similar, equal in this age pyramid, and the post-reproductive individuals are comparatively fewer in this age pyramid. So, okay, so that is why it is representing a stable age pyramid. Okay, so reason and assertion are both are true. Okay, and reason is the correct explanation of assertion. So, you can choose A here. Okay, A as a correct option for this assertion and reason type of question. Right. Then answer the questions from question number 17 to 21 and each carries 2 marks. Next we have 2 mark questions. Right. So let's see. A sequence is given here, CTTAAG and CTTAAG. What is the peculiarity of this sequence? Simply you can say. Right. So question is, what are such sequences called? Okay. So the sequence will be having a peculiarity that CTTAAG. So when we are reading from both the sides, it is somewhat similar. Okay. So these type of sequences are called as what? Palindromic sequences. Okay. So they are called palindromic sequences. Right. So, name the enzyme used that recognize such nucleotide sequences. For recognizing these kind of palindromic sequences, we can use endonucleases. Okay, so that is the enzyme. So, the enzyme responsible is endonucleases. It's a restriction enzyme, and this endonuclease enzyme will read the sequences inside within the DNA sequence. So that is why endonucleases is being used for the recognition of these kind of palindromic sequences. Then second part, what is their significance in biotechnology? Okay, for biotechnology, what is the significance of these kind of sequences? So in biotechnology, there is a restriction endonucleases enzymes are using. Okay, for cutting these kind of, and they will be making cuts in these regions. Okay, and they will make sticky ends. Okay, counterparts of sticky ends on both the ends. So we can ligate the foreign DNA and the plasmid DNA very easily by means of these sticky ends, which is creating by these enzymes. Okay, endonucleases. So this is the significance of these kind of sequences in biotechnology. Okay. Then question number 21, that is A part. Given below is a pyramid of biomass in an ecosystem where each bar represents the standing group available in the trophic level. Okay, and with the help of an example, explain the conditions where this kind of pyramid is possible in nature. So here you can see clearly that it's a pyramid of biomass, and here you can see the first trophic level will be here and the second trophic level. So the biomass will be different. Okay, so in the first trophic level, the biomass is less, and when compared to the second trophic level. Okay, so this is a kind of an inverted pyramid, right? So this is a kind of an inverted pyramid of biomass, which is an inverted pyramid of biomass. And here you can see in aquatic ecosystems, you can see this kind of inverted pyramid of biomass, right? So a small population, so you can explain with an example, right? So a small population, okay, of phytoplanktons will be representing in trophic level 1. Okay, phytoplanktons in aquatic ecosystem. Okay, phytoplanktons, and their biomass is very less, right? And these phytoplanktons, by depending these phytoplanktons, many fishes will be there, right? Fishes. So their biomass will be very large, right? So this is a kind of an aquatic ecosystem in which this is a kind of an inverted pyramid of biomass that is existing in aquatic ecosystem, right? And like that, in terrestrial ecosystem also, this kind of an inverted pyramid is being present. Okay, inverted pyramids are present. So this is actually an inverted pyramid of biomass representing small biomass phytoplanktons that occupying the first trophic level, and large population of fishes. Okay, their biomass will be very high when compared to the phytoplanktons. They are always depending on these phytoplanktons. Okay, next part B. Will the pyramid of energy be also of the same shape in this situation and give reason for your response. No, obviously we know that the pyramid of energy, which is always in upright condition, okay, which is always in an upright fashion. That means that the higher trophic level, the trophic level which will be having a greatest energy will be, how it will be like this, pyramid of energy will be always in an upright position. That is, here you can see the producers, okay, then that is the first trophic level, right, and this is the second level and this is the third level, that is the top carnivores will be present on the topest level, okay. So that producers will be having the highest amount of energy in this pyramid, okay, and coming from or moving from the first level to the second level, the energy will be somewhat low level, and again in the highest level, trophic level, again the energy will be very less in them, right. So this will be an upright fashion of a pyramid, okay. The pyramid of energy will be always in an upright fashion, okay. And give reason for your response. What is the reason for this? We know that is from each of the trophic level, that is energy flowing from the first level to the next trophic level, some amount of energy will be lost as heat, okay. You can expect some heat lost also. From the body of the organism, some amount of energy will be lost as heat, and the remaining amount of energy will be transferred to the next level. And from here also some amount will be lost as heat, and the remaining amount will be transferred to the next level, right. So this will be the fashion of this pyramid of energy, right. And there will be an OR question too. So, draw a pyramid of number where a large number of insects are feeding on the leaves of a tree, and what is the shape of this pyramid? Okay, so you can draw a pyramid, that is a pyramid of number, okay, pyramid of number which will be having a large number of insects, okay, that is feeding on leaves of a single tree, okay. So you can draw that the pyramid will be like this, okay, that is a single tree, okay, the tree will be here, okay, the leaf of a single tree, and depending upon this tree, you can find large number of insects, right? Large number of insects will be feeding on this tree, right? So the pyramid of number will be like this, okay. It is again an what? Inverted pyramid. Inverted pyramid of number, that is a single tree, leaf of a single tree, depending upon that tree, many of the insects are living, okay. Many of the insects will be feeding on the single tree. So this is a pyramid which is showing an inverted type of pyramid, which shows an inverted pyramid of number, okay. This will be the shape of this pyramid, inverted pyramid. And will the pyramid of energy be also of the same shape in this situation? Give reason for your response. No, okay. The pyramid of energy is always what? Upright, that is from one trophic level to the next trophic level, energy will be transferred, some amount of energy will be lost as heat, okay. And pyramid of energy will be always in an upright fashion. It can never be an inverted fashion, okay. So that will be the answer. Let's see, answer the questions from 22 to 28 question number, and each will carry 3 scores. That is next session, okay. And let's see question number 23. State the agents which helps in pollinating the flowering following plants. Okay, so you have given with the cone, water hyacinth and vallisneria. These are the plants given here, and you have to find out the agents that will helping in pollination in these plants. Okay, so explain the adaptation in these plants to ensure their pollination too. Okay, so first we have to take this corn plants. You know that corn plants. Okay, in corn plants, mainly of the flowers will be arranged as an inflorescence. Okay, and the tassels of the corn cob. Okay, they will be, they are the stigma, and their styles, the style of the corn. Styles are wavy. Okay, the styles of the corn plants are very much wavy according to the wind. Okay, and they will be collecting the pollen grains. Okay, that is the adaptation of corn plants for the pollination. So, wind is the agent of pollination in corn plants. Okay, according to the wind, the styles of the corn plants will get the inflorescence right? They will get wavy and they will collect the pollen grains. Okay, that is the method of pollination in corn. Okay, so that is wind is the agent. And in next one, that is in water hyacinth. Okay, in water hyacinth, the flowers will be present on the above the surface level of the water. Okay, so flowers will be present above the level of the water. Okay, so as like any of the land plants, the flowers that is above the water level, they will get pollinated with certain insects. Okay, insects or wind. Okay, so wind or insects may be the pollinating agents in water hyacinth plants. Okay, so the flowers will be present on the above level of the water. Okay, so as like any of the ground plants, okay, in land plants, the insects or winds will be acting as agents. Then in Vallisneria, water is the pollinating agent. Okay, water will be acting as a pollinating agent in Vallisneria, because in Vallisneria, the female flowers will be present on the surface of the water by means of long stalks. The female flowers will be having long stalks, and the female flowers will be present above the level of the water. Okay, or they will be present on the water, the female flowers, and the male flowers will be there, and the male flowers, okay, the small male flowers or the pollen grains from the male flowers, okay, they will be present in this water and they will be moving. The pollen grains will be moving according to the water current. The positive water current will move, helping for the movement of this pollen grains, and finally, eventually, some of the pollen grains, they will be reaching this female flower, okay, the stigma of the female flower, and they will be getting pollinated. Okay, so this is a method of pollination in Vallisneria. Okay, water is the agent, right? Then next question number 27. Carefully observe the given picture. Okay, this is an electrophoresis, right? So a mixture of DNA with fragments ranging from 200 base pairs to 2500 base pairs was electrophoresed on agarose gel with the following arrangement. Okay, here you can see the arrangement of agarose gel electrophoresis. Okay, you can see the positive and negative electrodes and the wells which will be having the buffers. Okay, DNA will be present in these wells, right? And question is, what result will be obtained on staining with ethidium bromide? Explain with reason. What you will observe or obtaining on staining the fragments of DNA with ethidium bromide? Okay, but carefully observe this figure once again. Okay, and you can find something that here the arrangement or the setup is will be a false one. Okay, the setup is not properly arranging the setup, so that we know that DNA fragments are negatively charged. DNA fragments are negatively charged. So, it cannot move from this positive end to the negative end or this negative electrode. Okay, so simply they are still present in the wells only in this setup. Okay, so this is the first condition. So, what will obtain on staining with ethidium bromide? You cannot obtain any bands. Okay, so no bands of DNA, because the DNA is negatively charged, it cannot move towards this negative end. Okay, they are only present in this wells. Okay, so that is the answer. And the second question, the above setup was modified and a band with 250 base pairs was obtained at X. Next we have, here you can see X. Okay, so the above setup was modified and we will obtain a band of 250 base pair at this X point. Okay, so the question is, what changes were made to the previous design to obtain the band at X? Okay, at this X position, you will be getting a band. Okay, so what changes were made to this previous design? What are the changes that you have to make? You have to change the positive and the negative terminal ends of the setup. Right, you have to interchange this terminals. Okay, here you can obtain the negative terminal and here you can see the positive terminal. So, the positive and the negative terminal ends, okay, this electrode should be interchanged. That is the change that we have to make here. Okay, and after that, when we have made that change, okay, here you can see the negative end and this is the positive end. Okay, then the DNA will start to move from this wells. Okay, they are moving and we obtain a band of 250 base pair at this X point. Okay, and why did the band appear at this position X? You know that DNA negatively charged, they are moving very fastly. Okay, so they are moving very fastly when small fragments, okay, they are moving very fastly. Okay, smaller fragments are moves fastly. Okay, smaller fragments of DNA, they will be move very fastly. They will be getting separated very fastly. Okay, so we'll get a band of 250 base pair at X. Okay, that is the answer of this question. Then, next, question number 28. There was a loss of biodiversity in an ecosystem due to a new construction project in that area. So, what would be its impact on the ecosystem? State any three of the impacts. Okay, so what is the impact on the loss of biodiversity in a particular area? Okay, the impacts of loss of biodiversity. What are the major impacts? You know that when a construction project will be going on a particular area. Okay, so in that area, we have to cut down many of the trees and plant population in that area for the construction processes, right? So, the plant population or the plant production in that particular area will get declined. Okay, so that is the first cause of concern or the first impact on the ecosystem. Okay, decline in plant production. The plant production in that area will get declined. That is the first impact. And secondly, what will happen? There will be an increased variation in certain ecosystems. Okay, increased variation in certain ecosystems. That means in certain, not all the ecosystem, in certain ecosystem we can expect more variations in that area. Okay, that is the another impact on the loss of biodiversity. Okay, and also there is a third impact. That means the resistance level of the of that particular area. Okay, the plant population in that area will get decreased. Okay, so decreased in the resistant level towards the environmental stresses like drought and all. Okay, when we are facing with that kind of environmental stresses, the resistance capacity will get decreased. Okay, so decreased resistance towards environmental stresses. Environmental stresses like drought and all. Okay, so these are the three main impacts on the loss of biodiversity in that particular ecosystem or that particular area. Next, list any three major causes of loss of biodiversity. Okay, the causes of loss of biodiversity. What are the major causes? You know that obviously habitat loss and fragmentation. Okay, that is the main cause of concern, right? So the first cause of loss of biodiversity is habitat loss. Okay, habitat loss and fragmentation. Then over-exploitation of certain species that is being already existed in that area. Okay, that is also the second cause of concern, right? So, over-exploitation. Then, we know that certain invasive species or alien species will be getting introduced into certain areas. Okay, they will get completely, the equilibrium of that ecosystem will get hindered with the introduction of the alien species. Okay, so introduction of alien species is another cause of concern. Introduction of alien species. Okay, then the last one is co-extinctions. Okay, co-extinctions of certain species. Okay, so these are the main causes of loss of biodiversity, main causes of concerns. Okay, then next one. Next session, that is question number 29 and 30 are case-based questions, and each question has subparts with internal choices in one subpart. Okay, and each of them carries 4 marks. Okay, and obviously in botany part, there will be no case-based questions in this question paper. Okay, so next session is answer the questions from question number 31 to 33, and each carries 5 marks. Okay, so question number 31. Trace the development of a megaspore mother cell to the formation of mature embryo sac in a flowering plant. Okay, it's a good question and very important one too. Okay, so the development of a megaspore mother cell to the formation of a mature embryo sac in flowering plants. Okay, we know that in flowering plants or in angiosperms, okay, the formation of an embryo sac is how that process will take place. Okay, so the process of formation of a megaspore from a megaspore mother cell. Okay, megaspore mother cell. We know that megaspore mother cell is also called as MMCs. Okay, megaspore mother cell. So from this MMC, what is being going to produce? That is the megaspores are going to produce megaspore. Okay, from megaspore mother cell, megaspores are forming. Okay, so the process of formation of megaspores from this megaspore mother cell, and this process is called as what? Megasporogenesis, right? So the process is called as megasporogenesis. Okay, megasporogenesis. And for this formation of these megaspores, or for the development of this process of megasporogenesis, what will happen actually? Okay, we know that in flowering plants, ovules are there. Okay, ovules. Okay, and inside this ovules, we have a large space, that is, it will be having a nucellus tissue inside it. Okay, a nucellus tissue. So inside the nucellus tissue, many of the cells will be present, and some of the nucellar tissues will be getting differentiated to form this megaspore mother cell. Okay, some of the cells of this nucellar tissue will be getting differentiated and it will be forming megaspore mother cell. Okay, and this megaspore mother cell, which is a cell, which is a larger cell, okay, which is a larger cell, which will be having denser cytoplasm, okay, dense cytoplasm and a nucleus inside it, okay, which is a normal cell. Megaspore mother cell is a large cell which dense cytoplasm and a nucleus inside it. Okay, and this megaspore mother cell undergo meiotic divisions. Okay, meiosis. It undergo meiotic division. Here you can observe the differentiation. Differentiation will take place to form megaspore mother cell, and this will be undergoing meiotic divisions. Okay, so megaspore mother cell undergo meiotic division and forming 4 haploid megaspores will be forming. Okay, 4 haploid megaspores will be forming after meiotic division, and out of these 4 megaspores, okay, out of these 4 megaspores, only one is functional. Okay, only one megaspore is functional. Only one is a functional megaspore, and the other 3 of the out of these 4 megaspores will get degenerated. Okay, 3 megaspores will get degenerated. Okay, 3 will get degenerated and only one is getting functional. Okay, only one is getting functional, and this one functional megaspore is being then in future they will be getting developed into a female gametophyte. A female gametophyte. Okay, that is, this female gametophyte is at last going to form this embryo sac. This female gametophyte in flowering plants or angiosperms is otherwise called as embryo sac. Okay, so this is the first differentiation or the first stages or the formation of this embryo sac or the functional female gametophyte from this megaspore mother cell. Okay, and the now the functional megaspore is forming the female gametophyte. Okay, so this functional megaspore, okay, it will be having a single nucleus inside it, and the nucleus in the next stage, the nucleus in the functional megaspore or the female gametophyte undergo mitotic division. Okay, so the functional megaspore, okay, the nucleus undergo mitotic division, mitosis. Okay, it undergo mitotic division and it will be forming after the mitotic division of the nucleus of the megaspore, okay, functional megaspore, it will be forming two nuclei inside it. Okay, so a functional megaspore which will be having two nuclei after the mitotic division of the single nucleus, and these two nuclei will be moving towards the opposite poles. Okay, they will be moving towards the opposite poles. Okay, after the first mitotic division. Then occurs two sequential mitotic divisions too. Okay, after this, two sequential sets of mitotic division will happen. After that, this 2 will be getting 4. Okay, then again a mitotic division will happens. Then it will be forming 8 nucleates. Okay, 8 nucleated condition of this female gametophyte or this functional megaspore. Okay, after 3 mitotic division, the single cell will be having 8 nucleus inside it. Okay, and these nuclear divisions are strictly nuclear. Okay, it does not following any cell wall formation. Okay, without cell wall formation, 8 nucleus will be present in the single megaspore. Okay, then after the 3 sequential mitotic divisions, after this 3 mitotic division, what will happens? The cells of this, the nucleus of these 3, out of this 8 nucleus, 3 nucleus will be moving towards the chelazal end. This is the chelazal end and this will be the micropylar end, micropylar region. Okay, 3 of the nucleus in the chelazal end, they will be getting followed by the cell wall formation. Okay, and they will be forming 3 cells at the chelazal end, and 3 cells will be forming at the micropylar end. Okay, they will be forming the egg apparatus. Okay, I will write here. Okay, that is inside this megaspore, what will happens? Out of the 8 nucleus, 3 of the nucleus will be on the micropylar end. This is the micropylar region and this is the chelazal region, chelazal region. Okay, and 3 of out of these 3 cells in the micropylar region, 2 will be forming synergids, synergids, and this is the egg cell. Okay, and altogether this is called as egg apparatus. Okay, egg apparatus is forming. Okay, so egg apparatus constitute 1 single egg cell and 2 synergids, and 3 of the nucleus in the chelazal end, they will be again forming cell walls surrounding it. Okay, and they will be forming 3 cells at the chelazal end, and these cells are altogether called as antipodals. Antipodals. And the remaining 2 nuclei, right? The remaining 2 nuclei will be positioned in the center. Okay, and this will be forming a central cell. Okay, this is the central cell and the cell will be having 2 nuclei in it. Okay, and these are called as polar nuclei. Polar nuclei or secondary nuclei. Okay, they will be forming the large central cell, forming inside this large central cell. Okay, so this is a functional embryo sac. This is a functional embryo sac in angiospermic plants, okay, or angiosperms, right? So, this is how from this megaspore mother cell, a complete embryo sac will be forming. And this embryo sac, there is a special feature in it, that is, it is a 7-celled, okay, which is a 7-celled, 8 nucleated embryo sac, okay, and this is a feature of an angiospermic embryo sac, which is 7 cells, 1, 2, 3, 4, 5, 6, 7, 7 cells, but it will be having 8 nuclei, 1, 2, 3, 4, 5, 6, 7, 8. 8 nuclei in it, right? So this is the feature, right? Then question number 33. Oil spill is a major environmental issue. It has been found that different strains of Pseudomonas bacteria, okay, they have genes to break down the 4 major groups of hydrocarbons in the oil. Trials are underway to use different biotechnological tools to incorporate these genes and create a genetically engineered strain of Pseudomonas, a superbug, to break down the 4 major groups of hydrocarbons in oil. So such bacteria might be sprayed onto the surfaces which is polluted with oil to clean thin films of oil. Okay, so this is a case. Okay, so based on this, we have certain questions. Okay, question A, list 2 advantages of using bacteria for such biotechnological studies. Okay, for such biotechnological studies, we are using certain bacteria. Okay, like here, in the previous case, Pseudomonas is a bacteria used for the particular controlling that oil spill. Okay, so what is the advantage of using these bacteria? Okay, so when we are using bacteria in biotechnological studies, it is very easy to culture these bacteria. Okay, so without any ethical issues, we can culture or we can cultivate a large number of bacteria. Okay, within the lab condition without any ethical issues. Okay, and these bacteria will get transformed very easily. The transformation of these bacteria is very easily too. Okay, very easy to conduct. So that is the main advantage when we are using this bacteria in biotechnological processes, right? Then for amplification of the gene of interest, PCR was carried out. We know that what is PCR? Okay, polymerase chain reaction with 3 major steps. Okay, what are the major steps of PCR? It will be having 3 steps, like the first step is denaturation. Okay, denaturation. Then we have annealing. Then the third step is extension, primer extension. Okay, so these are the 3 steps when we are utilizing PCR reaction. Okay, so amplification of that gene of interest, PCR was carried out in this particular case, and the PCR was run with the help of polymerase. Polymerase enzyme is using for PCR here, which was functional only at very low temperatures. This polymerase enzyme is functional only at very low temperatures. So how will this impact the efficiency of the PCR? Justify your answer. Okay, so we know that PCR is a reaction, polymerase chain reaction, for the amplification of a particular gene of interest or the fragment of interest. Okay, for this PCR is being carried out, and PCR will be following 3 major steps in it: denaturation, annealing and extensions. Okay, and here you know that in denaturation, we have to use a very high temperature for the denaturation of the DNA strands. Okay, we know that DNA is a double-stranded thing. Okay, so the double strands, the 2 strands will get separated to start the PCR reaction. Okay, and we are getting single strands each. Right, for the denaturation, we are using a very high temperature. Very high temperature we are using. Very high temperature. And then the strands of the DNA will get separated, right, after the denaturation step using high temperature. And then annealing, for this annealing we can use very low temperatures, not very low temperatures compared to the denaturation. Low temperatures are using. And again the extension step we are using again a high temperature. Right, so this is the temperature requirement for PCR reaction. Okay, but here in this reaction, we are using polymerase as an enzyme for the PCR to carry out. Okay, but the condition is that polymerase is an enzyme which will active only at a very low temperature. It is being functional. Okay, so we cannot use any low temperature for the denaturation, first step of PCR. Okay, so in that we cannot get any fragments. Okay, we cannot amplify the fragment of interest or the gene of interest. We cannot amplify. Okay, that is the condition. We cannot amplify the gene of interest. The gene of interest, because Now we are using polymerase as an enzyme here. Okay, this polymerase enzyme is active only at a low temperature. Okay, when we are applying very high temperature for the denaturation, the polymerase enzyme cannot withstand with that high temperature. Okay, so it will not give any amplification of the gene of interest. So that is the case. Okay, and if such bacteria are sprayed on water bodies with oil spills, okay, how will this have a positive or negative effect on the environment? Discuss it. Okay, so when we are will be having the water bodies with this oil spills, we are using this sprays of this bacteria. We are spraying these bacteria, Pseudomonas bacteria on the water bodies with this oil spills. Okay, that is a condition. And how will this have a positive or negative effect? Okay, we know that it will be having both a positive as well as a negative impact on that particular area. Okay, so first we have to discuss about the positive effects. What are the different positive effects that is being given when we are using this bacteria, okay, which is sprayed on water bodies with oil spills? What are the positive effects? When we are using these bacteria, what will be happen? The oil spills will be getting treated and the environment will become very safer and cleaner too. Okay, and the water is very potable for the aquatic forms that is present in the water body. Okay, the water is more potable for the aquatic forms. Okay, so that is the positive effect or the positive impact. And then it will be having the negative impacts too. Okay, what are the negative side of this condition, this situation? Okay, so when these bacteria, okay, they will conjugate with the other non-virulent forms, what will happen? They will be helping for the formation of another superbugs. Okay, that their effects, we cannot predict the effects of that superbugs. Okay, when they are conjugate with some other non-virulent forms, they will be there will be an unpredictable effect on the environment. Okay, we cannot predict what will be happens. Okay, we cannot predict that. So that is one negative impact. Okay, then when these bacteria they will be forming in the water body, they will be present in the water body, the dissolved oxygen level in the water bodies also will be very much decreased. Okay, so that will result in the mortality of the aquatic forms that is present on the water bodies too. Okay, so these are the negative effects, okay, when we are using this bacteria on the water bodies with oil spills, and positive effects too, right? Then in this session, we have an OR question too. Okay, let's see the OR question. Insects in the Lepidopteron group lay eggs on maize crops. Okay, so Lepidopteron insects, they will lay eggs on which crop? Maize crops. The larvae on hatching feed on maize leaf and tender cope. In order to arrest the spread of 3 such Lepidopteron pests, Bt maize crops were introduced in an experimental field. So, a study was carried out to see which of the 3 species of Lepidopteran pests was most susceptible to Bt genes and its product. The Lepidopteran pests were allowed to feed on the same Bt maize crops grown on 5 different fields, that is field A to field E. And the graph below shows the leaf area which is damaged by these 3 pests after feeding on the maize leaves for 5 days. Okay, let's see the graph first. Okay, so this is the graph given. So here you can see 3 species of Lepidopteran. 3 species of Lepidopteran. Okay, species 1 that is indicated in this dark shade. Okay, this is species 1. Then species 2. Okay, this light colored one, species 2, and species 3, this dark colored species, 3 species of Lepidopteran. Okay, and here in this graph, the x-axis is given with the leaf area which is damaged by these insect species. Okay, leaf area which is being damaged, and in y-axis given with the types of the maize in 5 different fields, field A, B, C, D and E. So, the 5 different fields, the type of maize is given in the y-axis and the leaf area damaged by the insects is given in the x-axis. Okay, so the question is, insect gut pH, there is a condition to here. So, the insect gut pH was recorded as 10, 8 and 6 respectively for the species 1, 2 and 3. Okay, all these species will be having their insect gut pH level of these insect species. In species 1, species 1, the pH is 10. In species 2, pH is 8. And in species 3, the pH is 6 in condition respectively. And the question is, evaluate the efficacy of the BT crop on the feeding habits of the 3 species of stem borer and suggest which species is least susceptible to BT toxin. Okay, which species is least susceptible? Okay, which is the least susceptible species? Let's see the graph once again. Least susceptible species, you can see that in species 3, okay, there is no damages in them, right? So, in first field, then second field, in third field, in field D, and in the last field 2, in species 3, okay, they are least susceptible. They are not getting damaged in these different fields when we are given that BT toxin 2, okay. So, the least susceptible species is species 3. Okay, they are very vigorously growing in these 5 different fields. Okay, so the least susceptible species is species 3. Right. Then, which species is most susceptible to the BT maize? Explain why. Which is most susceptible species? Okay, which species is most susceptible here? That is, here you can see that species 1. Okay, species 1 is present in field A, in 2, in 3, 4. But in the last field, we cannot find any species 1. Right? Is it true? Right. So, what is the reason for that? So, when we are applying the BT toxin to the insects, okay, the BT toxin is applicable, then what will happen? The BT toxin will be always in a protoxin form. Okay, the BT toxin, okay, when they are present first, they will be in a protoxin form. Protoxin means they are in an inactive form. They are in an inactive form, protoxin. Okay, only these toxins will be getting introduced into the insect's gut. Okay, then it will be getting activated. The active toxin. Will get the active toxin when only the toxin will be getting introduced inside the gut of the insects. Then only it will getting activated. Okay. So, in the last field, we cannot find any species 1 because the toxin will be getting more active in the species 1. Okay, in their gut epithelial cells, this toxins will get bind. Okay, then eventually the toxins will get activated in their pH conditions. Then what will happen? Cell swelling will happen and lysis of the cell will happen. Then eventually the death of that species will happen. Okay, the activation of this toxin inside the gut of the insects, right? So, we cannot find any species 1 in the last fields. Okay, very few in other fields, right? So, the most susceptible species, okay, the most susceptible species is species 1. Okay, the reason is also understood. And then, using the given information, suggest why similar effect was not seen in the 3 insect species. Okay, why similar effect was not seen in the 3 insect species? Why? Because we know that the pH level of these 3 insects will be getting different. Okay, they are different in condition. pH of the species 1 is 10 and the second species is 8. And these 2 pHs are alkaline pH. Okay, they are alkaline pH. Okay, so in the alkaline pH condition, what will happen? The crystalline protein will get solubilized in this alkaline pH. Okay, so the toxins will get activated. Okay, that is the protoxin, BT protoxin will get activated. They will be forming active toxin inside this alkaline pH. Alkaline pH, that is in 10 and 8 pHs. Okay, in species 1 and 2. So that the active toxin will give some changes to the species 1 and 2. Okay, their cells will get swells and lysis of the cell will happen and eventually death of that species will happen. But in the case of species 3, what will happen? It is the least susceptible species, species 3. Okay, that in the pH of this species 3. species 3 will be having a pH of 6. This is an acidic pH. Okay, this is an acidic pH. So, in this acidic pH content, the protoxin will not changes into its active form. Okay, they will remain in that inactive stage. Okay, then this toxin will not have any effect on the on their gut. Okay, so the species will not harmed with this toxin. Okay, because in that acidic pH, the protoxin will remain in its inactive form. It does not convert to its active form. Okay, only when they are converted into its active form, the changes will happen and the death of the species will ensure. Okay, otherwise the species will get flourished in the 5 different fields. Okay, that is why the species 3 will the species 3 will be found in the 5 different regions. Very flourishly they are growing because there is no effect on this toxin in that pH. Okay, in that acidic pH, the toxin will remain in that inactive form. So it does not cause any harm to that species. Okay, insect species. So that is the reason. Okay, in 3 different species, their pHs will be getting different. So that is why they will be having no similar effects. The effects will be are different types. Okay, so this is the question. So I think these are the different questions that we have to discuss here in this sample paper for your CBSE current pattern. Okay, so I hope you all understood. Okay, and thank you.